synthesis. So today we'll cover protein synthesis. In the previous class, we covered DNA replication. So before diving more into protein synthesis, we need to understand one of the facts, which is triplet code. Because so you need to remember this for a 2-mark definition. It usually comes what exactly triplet code is, and you need to answer this for 2 marks. Yeah. So basically, a triplet code is the coding of your nucleotide bases. And what are your nucleotide bases? We have 4 of them for DNA and 4 of them for RNA, right? They are nitrogenous in nature. We did adenine, we did guanine, cytosine, thymine. All 4 of them are present in DNA. And only uracil is one of the nitrogenous bases which you can find in RNA in place of DNA. Okay, so usually thymine is absent in RNA while uracil is only present in RNA. Okay, in place of thymine. So a triplet code is usually it's a sequence of your nucleotide nitrogenous bases which are present in our DNA, and they are responsible. They have this function. So if the question asks you to write the function of the triplet code, then you need to write this part of the definition. It is a code for the sequence of amino acid. So basically the triplet code, yani if there is 1 cytosine and 2 adenine, so this is a triplet code, and these 3 nitrogenous bases will be coding for a particular amino acid. Okay, so this is a code which what do I say? It codes for a particular amino acid. So we have 20 of them, right? We have 20 amino acids so far. So for example, if this is the example already given over here, CAA, it codes for the amino acid valine. Yani if CAA binds, so it will code for this amino acid valine, and then valine will be produced. So this is basically the code, and this is the actual product. And what exactly is the product? The product is your amino acid. And this code is always of 3 letters. So you need to remember this as well. Okay, why are we calling it triplet code? We are calling it triplet code because it has 3 letters in it. And then each sequence of 3 bases. So you can make multiple sequences, right? CAA, C double G, C double T, TTA. So anything. So you get multiple of sequences of the 3 bases, and each sequence of 3 bases will code for 1 amino acid. So if, for example, we have CAA, then it is only going to code for valine. It's not going to code for glutamic acid or any other amino acid like alanine or so on and so forth. So just remember this one example of CAA, that it's coding for amino acid valine. It's a good example, and you can write it in your answers as well. Okay? So when you're asked to write about triplet code, you need to tell that it is a 3-letter code, and these are responsible to code for amino acids. And then you can just give this example and you'll get 2 on 2 or you 3 on 3. So it depends on in the question as well. Okay, are they asking you about the function? Are they asking you about the structure? Or are they asking you about an example? So if they ask you this question for 3 marks, then you need to write all of the details, or else you can shorten it up if the question is of 2 marks. Now moving towards the important part, which is protein synthesis. So this is also a question which comes for 5 marks, and you need to write all of the information that is present in these slides because it's to the point. I have directly extracted it from your marking scheme, so do not try to shorten it up. Okay, all of this is present in your marking schemes. So Yani, technically, all of these are your keywords that should be a part of your script while you're writing about protein synthesis. So now protein synthesis means that you are now manufacturing a protein within your body. And who exactly is responsible? We have already talked about the fact that ribosomes are responsible for protein synthesis, or RER is responsible for protein synthesis. We already know about the background story, but now we are diving into the process of how it is actually made. So in the nucleus, part of DNA molecule unwinds and unzips. And when we have already talked about this fact, how DNA molecule unzips itself and between the hydrogen bonds are present, they are broken. Okay, so when they're broken, yani, as your double helix of DNA molecule unwinds or it unzips, the hydrogen bonds between the bases are broken. So we know, okay, between adenine and thymine, how many hydrogen bonds are present? 2 or 3?
2, miss.
2. And between cytosine and guanine?
3.
3. Excellent. So it's also written over here in my slide. आगे कोई भूल गया हो? So in adenine, when adenine and when thymine binds, or when they bond together, they form 2 hydrogen bonds in between, and cytosine and guanine will form 3 hydrogen bonds. Okay, so this is an important question. It usually comes in your MCQs. When we'll do past papers, you'll be able to understand it more, but abhi ke liye just remember this fact. Okay. All right. So the hydrogen bonds between the bases will break. Easy step. This is your first step. Okay. Then, as the RNA nucleotides pair up with their complementary ones, their sugar phosphate groups are bonded together by RNA polymerase to form a sugar phosphate backbone. So the important thing that now you need to remember, we talked about a phosphodiester bond in the previous part of the topic where we were talking about the DNA replication, that a phosphodiester bond is made. Now we are going to talk about a backbone, and that backbone is of sugar phosphate. We'll understand how a sugar phosphate backbone is made. Just hold on for a bit. We'll do it. The new single-stranded molecule which has formed is called messenger RNA. So one other step, right after the hydrogen bonds are broken, you need to remember that a single-stranded molecule is formed, and that single-stranded molecule is known as mRNA, and it leaves the nucleus via pore in the nuclear envelope. So you have RNA nucleotides inside your nucleus, and those RNA nucleotides, they are going to pair up with their complementary ones. We know how the complementary pairing takes place between the nitrogenous bases, right? We already know about this fact. And if you remember that you need to remember the structure of nucleotide, that it has a phosphate, then it has a sugar, then the 5-sided sugar, the pentose sugar, which is ribose in this case, and it has this nitrogenous base. Okay, so RNA nucleotides, they're going to pair up with their complementary ones, meaning if RNA has cytosine, then the complementary of cytosine is going to be guanine. So those two nucleotides are going to pair up with one another, and then their sugar phosphate groups are bonded together by RNA polymerase. So now comes the other enzyme that you need to remember. It has the same working as DNA polymerase. We did DNA polymerase for DNA replication. Now you need to remember RNA polymerase. Okay, sorry, that your nucleotide, we know it has a sugar and it has a phosphate. So the complementary nucleotides, they're going to bond. Their sugar phosphate bond is also going to be formed with the help of RNA polymerase. And then this part, the sugar phosphate part, is going to be binded with each other through RNA polymerase. Yani we know that nitrogenous bases are going to pair up in a complementary manner. Yani cytosine guanine paired up. Now, what about the sugar and what about the phosphate of each nucleotide? They are also going to bind with one another with the help of RNA polymerase. And once they bind with each other, once they bond with each other, they are going to form the sugar phosphate backbone. You know that when nitrogenous bases are pairing up with one another, they form hydrogen bonds, right? You're already aware about this fact. We have already written it over here as well. So now I'm just establishing one more fact. Okay, the nucleotides that have sugar and phosphate, when they are also supposed to bond with one another, and then they form the sugar phosphate backbone. So this is a very important word. Sugar phosphate backbone is formulated when complementary nucleotides, they pair up, and this particular one word is just a one mark when you'll write about protein synthesis. So just mark it with yourself that you need to establish another fact when you're talking about protein synthesis. A sugar phosphate backbone between the nucleotides is also formulated with the help of RNA polymerase. So these RNA polymerase and sugar phosphate backbone, they are of one mark, meaning that you need to write this in order to secure 2 marks. Okay? And then with this, when this process takes place, this entire process formulates a single-stranded molecule, and that single-stranded molecule is known as mRNA. So M stands for messenger. Okay? Yani, you need to remember this as well, कि mRNA, and remember to write it in a small manner. Yani, do not write this in a capital caps on log format. ठीक है? This is small m and capital RNA. Okay, do not make this mistake. They do not accept when you write all four letters into in a capital format. You need to small this up. Okay, small this up. Actually, so I'm saying कि the entire process which we have just talked about is formulating a single-stranded molecule, mRNA, and then this single-stranded molecule, it leaves the nucleus via a pore. It's a nuclear pore, ठीक है? In the nuclear envelope. So this entire process took place in your nucleus, and now when mRNA is formulated, it is going to leave the nucleus through the pore in the nuclear envelope. So far, are we good?
ब्लू बॉक्स अगेन थोड़ा सा समझा दें ब्लू बॉक्स
Blue box. अच्छा ठीक है यहाँ से समझते हैं हम. We said that in your nucleus your DNA is going to unwind, right? So this is, can you see this, that this is your DNA, double helix DNA, and you can see that it has nitrogenous bases, right? It has nitrogenous bases which are bonded with one another, and RNA polymerase is responsible to unwind it. Yeah, Lenny, we talked about DNA polymerase when we were talking about semi-conservative replication of DNA. So now I'm saying that RNA polymerase is going to perform the same function as DNA polymerase. It's going to unwind, it's going to unzip your DNA. ठीक है, जैसे जैसे DNA is passing through this RNA polymerase, it's going to unwind itself, and the hydrogen bonds between these bases are going to break. Now that we have this DNA strand, right? This is your DNA strand. ठीक है? Are you able to understand that this part is your DNA strand, which is unwinded? Are we good on this?
यस मिस
ठीक है. Now I am establishing another fact. Yani, this is your step number 1, that you need to unwind your DNA. Step 1 has taken place over here. Now the second part that I'm trying to establish is that in your nucleus there are free RNA nucleotides. Yeah, just we have free DNA nucleotides. Similarly, we have free RNA nucleotides as well in the nucleus. So far, good? समझ आई ये बात?
जी मिस
ठीक है. Those nucleotides have this structure, right? This is your phosphate, then a 5-sided sugar. 1, 2, 3, 1, 2, 3. ज़्यादा हो गई. I'm not sure कैसे बनती है. ऐसे, yeah. 1, 2, 3, 4, 5, yeah. And this is your nitrogenous base. ठीक है? This is your RNA nucleotide, which is present in your nucleus. समझ आई? This is your free RNA nucleotide. Just stop me at the point where you are unable to understand. Okay, these are your free
ओके
nucleotides present in the nucleus. Okay, they are present in your nucleus. Acha. Now I'm establishing this fact that this nucleotide will have particular nitrogenous base, obviously. So let's consider that this is your cytosine. Okay, this RNA nucleotide has cytosine. So cytosine will go over here and it will find guanine on the template strand of DNA. And let's assume that this part of this, this nitrogenous base is guanine. Okay, वो cytosine थी. अब if this is guanine, then this RNA nucleotide is going to come over here. It's going to attach itself, and the cytosine part will form a bond with guanine. Make sense?
यस
ठीक है. अब since this is not a 3-dimensional picture, but another fact that I'm trying to establish is कि अब इनकी तो bonding हो गई ना, यानी your nucleotide, your nitrogenous bases are binding with one another with the help of hydrogen bonds. Another fact that I'm trying to establish is कि RNA nucleotide has this sugar phosphate, right? This sugar phosphate. So since this is not a 3-dimensional picture, I'm trying to say that DNA का sugar phosphate and mRNA का sugar phosphate, it's going to form a bond, and that is going to be sugar phosphate backbone of mRNA. ठीक है, अब this is not a 3-dimensional picture, वरना ये थोड़ा twisted होती, but just थोड़ा सा विजुअल इफेक्ट से समझने की कोशिश करें, यू विल नॉट बी एबल टू फाइंड अ पिक्चर ऑफ इट। या मैंने फिर आपको विजुअली समझना पड़ेगा कि ये वाला जो पार्ट है जो थोड़ा डार्कर साइड है, इट्स द शुगर फॉस्फेट बैकबोन ऑफ mRNA। यानी ये आपका डीएनए अनवाइंड हो गया, एंड बीच में आपका आरएनए न्यूक्लियोटाइड्स दे आर बाइंडिंग विद द टेम्पलेट स्ट्रैंड ऑफ डीएनए। जहाँ पे ओपन अप हुआ वहां पे वो अपने आप को स्टिच करवाते हैं, एंड पीछे जो शुगर फॉस्फेट पार्ट है, शुगर फॉस्फेट पार्ट क्या बना रहा है, बैकबोन बना रहा है mRNA की। और जैसे जैसे ये बनती जाती है, जैसे जैसे न्यूक्लियोटाइड्स कनेक्ट, यानी अभी आई एम सेइंग कि ये जो न्यूक्लियोटाइड इधर है, इट्स नॉट इन अ composed manner, it's free in the nucleotide, but now when it is stitching itself, मैं इसको स्टिचिंग इफेक्ट बोलती थी बिकॉज मुझे ये स्टिच लगता था कि अगर आपने सिलाई मशीन देखी हो, उसमें नीडल होती है एंड उसमें से कपड़े पास करते हैं, तो मुझे ये कपड़े लगते थे और यहाँ पे मैं इसको नीडल बनाकर समझती थी। सो दैट वाज माय वे ऑफ अंडरस्टैंडिंग इट। इसलिए मैं स्टिच का वर्ड बार बार यूज कर रही हूँ बिकॉज इट्स हाउ आई लर्नड इट। डू नॉट राइट द वर्ड स्टिच। ठीक है, सो अब यहाँ पर विद द शुगर फॉस्फेट बैकबोन, these RNA nucleotides, they have attached itself, right? They are no longer free. They are in an attached manner. And once they are attached, and once the sugar phosphate backbone is made, they leave the nucleus through the nuclear pore via nuclear envelope. Okay, so this single-stranded part, this single-stranded part of mRNA has been formulated, and now it's leaving the nuclear pores via nuclear envelope. Okay, it's moving towards the cytoplasm part. Have all of you understood now?
Yes, miss.
Okay, excellent. I know it's hard, but ho jayega, don't worry. Acha, then in the cytoplasm, so now this is the third step. In the cytoplasm, there are free amino acids. There are free amino acids and another RNA molecule, which is transfer RNA. So you need to remember that tRNA, you are going to write in the same manner like I taught you to write mRNA. Okay? Write T in a small manner and RNA in a capsule manner. Okay? So in your cytoplasm, there are free amino acids and tRNA molecules. From nucleus, mRNA is coming. In cytoplasm, tRNA is present and free amino acids are present. At one end of each tRNA molecule is a site to which an amino acid can bind. One thing that you need to remember, I'll show you how tRNA looks. And at the other end are three unpaired bases. The base triplet is called an anticodon. So the codon we just studied, the triplet code is already there. So the triplet code is known as codon, while the one which is present in an unpaired base manner on the tRNA, it's an anticodon. I know you are unable to understand this, but just hold on to the word anticodon. I'll show you the diagram. Don't worry about it. And then each tRNA molecule bonds with a particular amino acid under the control of specific enzyme and with the energy from ATP. So now let's study how tRNA looks. This is your tRNA. Okay? This is how your tRNA looks. This part is tRNA. Okay? We did that tRNA has a site of attachment for amino acid. So this is the site for the attachment of amino acid, and the other end of tRNA has three bases which are in an unpaired manner. And these three bases, for example, if this is AUG, for example, if this is AUG, and why I'm writing AU, I'm writing U because this is a RNA. This is a tRNA, meaning it's an RNA. So we'll not write T, we'll write U. So uracil is present in RNAs, okay? Remember this. So for example, AUG is present over here. So I'll establish the other fact later on, but just understand this fact. This is now these unpaired bases present on the tRNA. These are known as anticodon. Okay? These are anticodon, and the one which will be arriving with the help of mRNA, those are going to be known as codons. Okay? So tRNA, let's just do the structure of tRNA and then we'll study more. Okay, tRNA has a site of attachment for amino acid, and the other end of tRNA has three unpaired bases, and those three unpaired bases are known as anticodon. Just learn this for now, and let's take a 10-minute namaz break. We'll cover the rest in the second part of the class, okay? I'll open the classroom at 6:06.